Proteases are one of the most investigated groups of biomolecules both in academic and industrial research. Their pivotal role in several physiological and pathological processes makes them a prominent target in the field of drug discovery as well. Regarding their intensive research, there is a great and continuous demand for the development of time and cost-saving, high-throughput screening-compatible fluorescent protease assay platforms. Here, we would like to introduce a protocol for the application of a separation-based, high-throughput screening-compatible fluorescence assay platform using both mTurkis2 and M-Apple-fused recombinant fusion protein substrates. Additionally, we would like to demonstrate a method for the in-gel renaturation of the substrates and the cleavage fragments of the assay samples after denaturing SDS speech as well. All the experiments are carried out on the example of HIV-1 virus protease. The expression plasmid carries the coding sequence of a hexahistidine affinity tag and a maltose-binding fusion protein, followed by a control cleavage site of tobacco H virus protease, a cloning cassette, and the C-terminal fluorescent protein. Linearize the expression plasmid by PEC1 and NAG1 restriction endonucleases. Perform the annealing of the Escherichia coli codon-optimized forward and reverse oligonucleotide primers that code for the substrate sequence of interest and flanked by PEC1 and NAG1 cohesive ends. Perform the insertion of the annealed primers into the linearized expression plasmid by ligation. For protein expression, transform Bia21DE3 competent cells by the ligation mixture. The fluorescent proteins will be in the same open reading frame with the N-terminal